DNA replication. DNA is replicated in a manner called semi-conservative replication. Semi-conservative replication produces two copies that are each half of the daughter strand and half newly synthesized molecules.
DNA is structured as a double helix with deoxyribose sugars, phosphates, and nitrogenous bases intertwined about a common axis.
The backbone of DNA is made up of alternating phosphates and deoxyribose sugars with nitrogenous teeth.
These teeth are actually bases, and there are four different types. These are adenine, thymine, guanine, and cytosine.
These bases each join together in a certain manner, with pairs of two being made. Adenine binds to thymine, while guanine binds to cytosine. There is no other way for the nitrogenous bases to be bonded.
These bases and sugars are all part of a complex called a nucleotide. Nucleotides are organic molecules that serve as the units for forming the nucleic acid polymers of deoxyribonucleic acid.
As DNA is being replicated, it is going to begin at the origin of replication, causing the DNA to split into two strands, leading and lagging, forming a replication bubble. DNA polymerase synthesizes continuously along the leading strand because of its 5' to 3' directionality.
Another strand is also created and is called the lagging strand, which has 3' to 5' directionality.
Because of its 3' to 5' directionality, the lagging strand is built in a series of fragments called Okazaki fragments, which we will get to in a little bit.
For the step-by-step procedure of DNA replication. The first step of the replication process occurs when an enzyme called topoisomerase comes along and helps to, in a sense, flatten out the strand of DNA from a double helix to a ladder in order for the rest of the process to occur easier.
Next, an enzyme called DNA helicase untwists the double helix at the replication fork. This splits the DNA into two strands, the leading and the lagging strand, as discussed before.
A protein called the single-stranded binding protein is used to help keep both the leading strand and the lagging strand separate from each other. RNA primer is then laid down by primase for DNA polymerase to act upon.
An enzyme called DNA polymerase now can come along and attach new bases to match the daughter strand according to the complementary base pairings of A to T and G to C. Polymerase proofreads the DNA strand and helps correct any misplaced nitrogenous bases. Additionally, it removes the RNA primer and removes it and inserts the nucleotides.
Due to the lagging strand's 3' to 5' directionality, the lagging strand is replicated differently. DNA polymerase can only work from 5' to 3', so primase lays down RNA primers in pieces called Okazaki fragments.
The RNA primers are removed and filled by DNA polymerase, and the fragments are sealed together by ligase. Because of its 3' to 5' directionality, the lagging strand has to be made in these Okazaki fragments. DNA polymerase can only work from 5' to 3'. This means that in the leading strand, DNA polymerase can act continuously.